It's almost unsettling, really, when you think about it. A condition affecting so many people, hundreds of millions globally. And it's behind such serious health events, but it often just creeps along silently for decades. Today, we're kind of pulling back the curtain on this quiet giant.
We are. We're doing a deep dive into the underlying mechanisms of atherosclerosis.
Exactly, atherosclerosis, yeah. And our insights today, they come from a pretty detailed chapter in a medical textbook.
Yeah.
Think of this as, your shortcut to understand the core stuff.
But explained in a way that hopefully sticks.
Right.
Our goal isn't to get bogged down in every single tiny detail, but to really get a handle on what atherosclerosis is, how it sort of establishes itself in our bodies.
And why it matters so much.
Exactly, why it's so critical for our long-term health. We're here to pull out the most important and, frankly, the most fascinating bits.
Okay, let's unpack this then. Right off the bat, the chapter hits you with the sheer scale of it all. Atherosclerosis-related cardiovascular disease, CVD.
Yeah.
It's not just a health concern.
No, it's the leading cause. Death and disability right across the globe. It really does make you stop and think.
And just to frame that, you mentioned ischemic heart disease and stroke.
Yeah, often the direct results of atherosclerosis. They're #1 and #3 causes of death worldwide. Think about that.
Wow.
Together, they account for something like 84.5% of all cardiovascular deaths and over 28% of all deaths from any cause.
It's a massive figure.
It really is.
But what's interesting, though, is the chapter mentions that while the rate of people dying from CVD has actually gone down a bit.
Likely due to better treatments, yeah.
Right. But the number of people living with it is actually going up, which, of course, creates this huge financial burden.
Oh, absolutely. The economic impact is just immense. In the U.S. alone, estimates are over 120 million people have some form of CVD.
120 mil?
And the annual cost, over $350 billion, just for treatment.
Incredible.
And globally, you have conditions like peripheral arterial disease, PAD, also linked to atherosclerosis, affecting over 200 million. And it hits lower-income countries harder, disproportionately so. It's a truly global health challenge.
But here's the part that really got me, and it's, well, a bit concerning. The chapter highlights how common the risk factors are, even in kids, young adults.
Yes, this is so important.
Autopsy studies have actually shown a direct link between things like, sedentary lifestyle, abdominal obesity, poor diet leading to bad cholesterol, high blood pressure.
All those things we hear about.
And the very early stages of these lesions forming in arteries, it really makes you think about modern lifestyles,
It's a critical point. Studies like the Bogalusa heart study and the PDAY study, that's pathobiological determinants of atherosclerosis in youth, they were pivotal.
Right.
They showed these atherosclerotic lesions, they can start forming way back in childhood, and then they just get progressively worse with age. It's like rust on a new car. Starts small, unnoticed.
For years later.
Exactly. The damage can be significant, even irreversible.
And it's not just tiny microscopic things, is it? There was that IVUS study, intravascular ultrasound.
That's right, looking inside the arteries of heart donors. They found evidence of atherosclerosis in 17% of people younger than 20.
17% under 20. Wow, that's sobering. These changes happening without any outward signs at all.
And what's fascinating, or perhaps alarming, is the clear link between these early lesions and the known CVD risk factors. Things like familial hypercholesterolemia, that's inherited high cholesterol hypertension, severe obesity, type 2 diabetes.
In childhood?
Yes, in childhood. They strongly correlate with developing these lesions early and with how severe they are. Plus, other conditions like type 1 diabetes, chronic kidney disease, chronic inflammation, they all play a role.
It really drives home the importance of, early risk assessment and those lifestyle changes we always talk about.
Absolutely. The chapter even mentions seeing diffuse intimal thickening, an early sign in coronary arteries as early as 36 weeks of gestation.
Before birth.
Essentially, yes. In those areas prone to atherosclerosis later.
So the story starts incredibly early, often completely silently. What's the next step? How do these first signs, these fatty streaks, how do they turn into something more serious?
Well, the progression leads to what we call advanced lesions or fibrous plaques. These are different. They've got a significant buildup of extracellular lipids, fats outside the cells, and also fibro-connective tissue.
Okay.
They tend to look whitish, and they actually bulge into the artery's lumen, the channel where blood flows. So you can immediately see how that might start to narrow things down.
And these are the ones that cause the real trouble.
Right.
The ones leading to heart attacks, strokes.
Precisely. These fibrothromas, that's a technical term, can cause problems in a few main ways. The lining, the endothelial cells, can wear away, that's erosion.
Okay.
Or the fibrous cap, like a protective cover over the plaque, can rupture. That's a big one.
Rupture. Sounds bad.
It is. Or you can get erosion of a calcium nodule within the plaque, or even bleeding inside the plaque itself, intraplaque hemorrhage. Any of those can kick off a really dangerous cascade.
And the chapter mentioned these plaques aren't all created equal. Some are soft, some hard.
Exactly. They vary quite a bit. Some are soft, more friable, easily broken. Others are dense, heavily calcified. And that fibrous cap you mentioned is really crucial.
How so?
Well, it acts like a shield over the plaque's core. This core, the necrotic core, is usually deeper inside. It contains this sort of, well, the book calls it porridge-like gruel of lipids and cholesterol crystals. Atheroma literally means porridge.
Porridge.
Okay, and the fibrous cap is made of smooth muscle cells and collagen. But, and this is key, in plaques that are vulnerable, prone to rupturing, this cap can become very thin. It gets infiltrated by inflammatory cells, especially macrophages.
Inflammation again.
Yes. Identifying these thin cap fibrothromas is a huge focus of research right now, using advanced imaging.
So we've got these lesions developing sometimes silently for ages. What are the big ideas about how this whole process actually gets started? The chapter laid out a few key theories, didn't it?
It did. You've basically got the lipid hypothesis, the response to injury hypothesis, and the monoclonal hypothesis. Let's start with the lipid hypothesis. That one's been hugely influential for a long time.
Right. This goes way back, doesn't it? To Inichko and his rabbits.
Yeah.
Early 20th century.
That's the one. He showed feeding rabbits cholesterol caused vascular lesions that looked remarkably similar to human atherosclerosis, a groundbreaking finding at the time.
Suggesting high cholesterol was the main driver.
Exactly. Then came the discovery of lipoproteins. Think of them as cholesterol taxis in the blood. And the key insight wasn't just total cholesterol, but the type of lipoprotein carrying it. VLDL, IDL, LDL, HDL.
LDL being the bad one, HDL the good one, generally speaking.
Right. And a protein called ApoB100 is found on those bad carriers, VLDL, IDL, and LDL. It became a marker.
And then the huge breakthrough, Goldstein and Brown, right? Nobel Prize work.
Absolutely pivotal. Their discovery of the LDL receptor and how defects in it cause familial hypercholesterolemia, incredibly high LDL levels that really cemented LDL's role.
It showed how cells take up LDL.
And without that receptor?
LDL just builds up and up in the blood. That work, plus large population studies like the Japanese migration studies, and of course, the Framingham Heart Study.
Tracking people for decades.
Yes, clearly linking cholesterol levels to CVD risk. And then clinical trials sealed the deal, showing that lowering cholesterol actually reduced cardiovascular events.
So the lipid hypothesis, lipids, especially LDL, are critical, okay? But then there's the response to injury hypothesis. What's the thinking there?
This theory shifts the focus slightly. It suggests the process starts with some kind of damage or injury to the endothelium, that smooth inner lining of the artery.
Damage from what?
Could be mechanical stress, like from high blood pressure, toxins from smoking, maybe infectious agents or inflammatory signals. Think of it like getting a scratch on the inside of the pig.
Okay, so there's an injury. Then what? Well, that injury allows platelets, the clotting cells, to stick to the damaged spot. It also lets LDL and other stuff from the blood seep into the layer just beneath the lining, the subendothelial space.
Right.
Then these platelets release growth factors that call in smooth muscle cells from the artery wall. These cells migrate in, they multiply, and they start laying down matrix material, thickening the wall. It's like a repair process gone a bit haywire.
A chain reaction, basically. But there was also a counter idea mentioned, response to retention. How's that different?
It's a subtle but important difference. The response to retention idea suggests the first critical step isn't necessarily endothelial injury, but rather the trapping, the retention of those ApoB lipoproteins, like LDL.
Getting stuck under the lining.
Exactly. Getting stuck in the subendothelial space, specifically in atherosclerosis-prone areas. This retention, even before you see fatty streaks, is what triggers the inflammatory response from macrophages and T cells. So lipids getting stuck comes first, then the inflammation ramps up.
Interesting distinction. Okay, what about the third one, the monoclonal hypothesis? That sounded different again.
It is quite different. This idea, mainly from the Bendits, proposed that each plaque doesn't just arise from a general process, but actually starts from a single, smooth muscle cell.
One cell.
One cell that starts dividing uncontrollably, expanding clonally. So all the smooth muscle cells in that specific plaque are descendants of that one original mutant cell.
And they had some clever genetic evidence for this using an enzyme, G6PD.
Exactly. In females, one X chromosome is inactivated in each cell. G6PD is on the X chromosome. They looked at black females who were heterozygous for different G6PD types. They found that within a single plaque, the smooth muscle cells usually only expressed one type of G6PD.
Whereas the normal artery tissue next to it showed a mix of both types.
Precisely, suggesting they all came from one parent cell with one active X chromosome.
That's pretty elegant detective work, even if there are debates about it. It pointed towards a potential genetic or cellular trigger, right?
Yeah. It certainly did. And really, all these hypotheses have sort of fed into our current understanding, which sees atherosclerosis as fundamentally a chronic inflammatory disease. It's not just plumbing clogged with fat.
Right. The chapter really hammers this home. The immune system, both innate and adaptive, is involved in pretty much every step.
Absolutely. It's a dynamic biological process. Animal models have been crucial here, especially mice engineered to lack ApoE or the LDL receptor.
The ones that get really high cholesterol and human-like plaques.
Yes, and when you cross these mice with mice lacking parts of their immune system, they develop significantly less atherosclerosis. That really underscores immunity's role in driving the disease.
So let's dig into the immune players. LDL retention comes up again, but in the context of inflammation now.
Yes. As we said, those ApoB lipoproteins get trapped in the subendothelial space. Once there, they get modified, oxidized, essentially, like metal rusting.
And that oxidation is like a red flag.
Exactly. Oxidized LDL is highly chemotactic. It attracts monocytes, a type of white blood cell. The activated endothelium also puts out adhesion molecules, like Velcro.
To grab the monocytes floating by.
Precisely. Grabs monocytes and lymphocytes, then chemical signals, chemokines, lure these monocytes into the subendothelial space. It's a targeted recruitment.
And once inside, the monocytes change into macrophages, the Pac-Man.
Correct. And these macrophages start gobbling up the oxidized LDL using special scavenger receptors like SRA and CD36. As they fill up with lipid, they transform into foam cells.
The hallmark cells of these plaques.
That's right. And factors like macrophage colony-stimulating factor push this process along, making macrophages multiply and release more inflammatory signals, or cytokines.
And there are different types of macrophages involved, like pro-inflammatory M1s.
Yes, under high lipid conditions, these M1s become dominant. They churn out key pro-inflammatory cytokines like IL-1 beta and TNF-alpha. Think of these as alarm bells and instructions for the immune response.
And blocking these signals can help.
Studies show blocking IL-1 beta, for instance, reduces lesion size in animal models. IL-18 is another important pro-inflammatory player.
So these macrophages aren't just sitting there full of fat, they're active participants.
Very active. They're abundant in ruptured plaques. They produce enzymes called matrix metalloproteinases, MMPs.
The ones that break down the cap.
Yes, they degrade the collagen in the fibrous cap, weakening it. Macrophages also express tissue factor, a potent clotting trigger. This directly links the inflammation to the risk of thrombosis blood clots, hence the term atherothrombosis.
Okay, that's the innate system. What about the adaptive immune system? T cells, B cells?
Well, seeing macrophages displaying MHC class II molecules, those are involved in antigen presentation, hinted that T cells were involved. And sure enough, T lymphocytes make up 10–20% of the cells in lesions.
And they're active.
They show signs of chronic activation, producing inflammatory signals like interferon gamma. Remember those immune-deficient mice? SCID mice lack T and B cells. Crossing them with atherosclerosis-prone ApoE knockout mice leads to a dramatic reduction in lesions.
Wow.
And if you transfer T cells specifically reactive to oxidized LDL back into these mice, lesion formation accelerates. It clearly shows adaptive immunity is playing a role, likely recognizing modified self-antigens.
Like the body attacking itself. The chapter mentioned autoimmune parallels.
That's a good way to think about it. Potential autoantigens could be modified LDL itself, or maybe heat shock protein 60. Different T cell subsets seem to have different effects, some promoting, some maybe regulating the inflammation. Even mast cells found at the plaque shoulders might contribute to rupture.
It's incredibly complex. What were the smooth muscle cells again? We talked about them originating the plaque maybe, but what's their role in this inflammatory soup?
Well, those inflammatory signals we discussed, they stimulate smooth muscle cells to migrate from the middle layer of the artery wall into the inner layer.
Okay.
Once there, they proliferate, driven by growth factors.
Yeah.
And crucially, they produce collagen and other matrix components. They build the fibrous cap, essentially trying to wall off the inflammation.
But sometimes that cap isn't strong enough, or the cells themselves change.
Right. In advanced lesions, these smooth muscle cells can become senescent, less functional, and they can also be involved in calcification.
Ah, yes, the hardening of the arteries. How does that happen?
A molecule called BMP2, often released by stressed endothelial cells, along with phosphate, can trigger smooth muscle cells to change into bone-like cells, leading to calcium deposits. Why some plaques calcify heavily and others don't isn't fully clear, but it's part of the process.
So it's this whole mess.
Yeah.
Lipids get trapped, the immune system reacts, tries to clean up but causes more inflammation. Smooth muscle cells try to repair and contain it, and this can all go on silently for decades.
That's a pretty good summary. And a key piece linking the lipids to the intense inflammation is something called the inflammasome, specifically NLRP3.
The inflammasome.
There it is, that too.
It's a protein complex inside cells. And crucially, it can be activated by cholesterol crystals forming within the plaque.
The cholesterol itself triggers it, yes. This activation leads to the production of caspase-1, an enzyme that then activates potent cytokines like IL-1 beta. So it's a direct link. Cholesterol crystals, inflammasome, IL-1 beta, more inflammation.
And there was clinical evidence for this, the CANTOS trial.
Exactly. That trial tested an antibody-blocking IL-1 beta, canakinumab, and it significantly reduced cardiovascular events in high-risk patients, proving this inflammatory pathway is clinically relevant in humans.
Fascinating. And that brings us to C-reactive proteins, CRP. We hear about that a lot.
Yes, CRP is a well-established biomarker of systemic inflammation. Numerous studies linked higher levels of hsCRP, high-sensitivity CRP, to increased risk of heart attacks, strokes, PA progression.
Sometimes even more strongly than LDL cholesterol.
In some studies, yes, it adds independent prognostic information, which is why it's often included in risk assessment tools.
And the Jupiter trial was key here, wasn't it? People with normal LDL, but high CRP.
Right. It showed that treating those individuals with a statin, rosuvastatin, significantly reduced their cardiovascular events. It really highlighted inflammation's role, even when cholesterol isn't overtly high, and suggested statins have anti-inflammatory benefits beyond just lowering LDL.
Okay, so we have a much clearer, albeit complex, picture of how these plaques develop. But why do they prefer certain spots in our arteries? Not just anywhere.
That's a really interesting aspect. Atherosclerosis definitely has favorite locations, and it relates strongly to blood flow dynamics or hemodynamics.
So how the blood flows matters.
Immensely. Areas with smooth, steady, laminar shear stress, think of smooth flow along a straight pipe, tend to be protected. They're resistant.
Okay.
But areas with turbulent flow or low shear stress, like at branches, bifurcations, the inside curves of arteries, those are the susceptible spots, like the carotid bifurcation in the neck.
Why the difference? What does smooth flow do?
Laminar shear stress is actually good for the endothelium. It stimulates the production of nitric oxide, NO.
Nitric oxide.
Yeah, NO is protective. It helps prevent clots, reduces those adhesion molecules that grab immune cells, inhibits smooth muscle cell migration, and even boosts antioxidant defenses.
So smooth flow keeps the lining healthy and non-sticky.
Basically, yes. Turbulent flow, on the other hand, disrupts this. It might increase the residence time of particles like LDL near the artery wall, making them more likely to get trapped. The physical forces literally change gene expression in the endothelial cells.
Amazing. Physics influencing biology at that level. It explains why some areas resist plaque, even with risk factors present.
Exactly. And it also ties into the idea that these plaques aren't static. They're dynamic. They grow, they might stabilize, they might even shrink sometimes.
Right. The chapter talked about growth spurts and quiet periods. What causes those shifts?
Well, one thought is that small, clinically silent plaque disruptions, tiny erosions or ruptures with minimal clotting might happen frequently. Each time, there's a local healing response, inflammatory signals, growth factors, which can actually lead to gradual plaque growth and narrowing over time. It's not always one big bang.
Little injuries adding up. And this explains why standard angiograms can be misleading. An artery might look okay,
but have significant plaque hidden in the wall. That's where IVUS, the intravascular ultrasound, is so revealing. It shows the artery often remodels, expands outwards initially to accommodate the plaque. Lumen narrowing is actually a late event.
so the pipe gets wider before it gets narrower.
In many cases, yes, compensatory enlargement. But what's also encouraging is that IVUS studies like reversal and asteroid showed that with really intensive statin therapy.
Lowering LDL way down, reducing CRP.
Exactly. You can actually see plaque regression. The plaques can shrink. These trials strongly support aiming for lower LDL targets.
So it's not necessarily a one-way street. Aggressive treatment can make a difference.
It absolutely can. But of course, the major concern remains the acute thrombotic complications, the sudden blood clots.
The heart attacks and strokes. And you mentioned it's not always the tightest blockages that cause them.
That's a key point. A very severe stenosis, say 90% blocked, carries a high risk for that specific lesion. But there are far more lesions causing only moderate narrowing, maybe 50% or 60%.
Right.
So statistically, more heart attacks actually originate from these less severe but much more numerous plaques. You have to consider the total number of potential culprits.
So it's less about the degree of narrowing and more about the stability of the plaque, its vulnerability.
Precisely. It's usually a physical disruption of the plaque that triggers the clot. The chapter outlines 4 main ways this happens.
Okay, what are they?
Number 1, fracture of the fibrous cap. This is the most common cause of fatal coronary thrombosis.
Okay.
Number 2, plaque rupture. This involves both mechanical stress and biological factors, things like a large lipid core, a thin cap, vulnerable spots often at the shoulders of the plaque.
Makes sense.
Number 3, superficial erosion of the endothelial lining. Just the surface layer wears away, exposing the underlying tissue. This seems more common in diabetes and in women.
Interesting.
And number 4, erosion of a calcified nodule that protrudes into the lumen.
And inflammation is weakening the cap in many of these cases.
Absolutely. Those macrophages and mast cells releasing MMPs, degrading the collagen matrix, inflammatory signals driving tissue factor expression, priming the clotting cascade, it's all linked.
So the cap breaks or erodes, blood hits the stuff underneath.
And that stuff is highly thrombogenic clot promoting. Tissue factor kicks off the clotting cascade, platelets pile on, boom, you get a thrombus, a clot.
Which can block the artery.
It can block it completely, causing a major event, or partially block it, leading to unstable angina or further stenosis progression. And factors like smoking, high blood sugar, high LDL, they make the blood itself more prone to clotting too.
The chapter also mentioned bleeding within the plaque, intraplaque hemorrhage.
Yes. Especially in carotid plaques, this can cause symptoms. New, fragile blood vessels can grow into the plaque neovascularization, and these can easily rupture and bleed.
And the type of complication might vary depending on the artery.
It seems so. Coronary and carotid plaques often have larger lipid cores, more prone to rupture. Leg artery plaques, femoral plaques, tend to be more fibrous, maybe more prone to erosion.
And the fact that anticoagulants, like factor Z inhibitors, show benefit in trials like Compass and Voyager.
It strongly supports the role of thrombosis, of clotting, as a key driver of adverse events in people with atherosclerosis.
So given all this complexity, how on earth do we identify these vulnerable plaques before they cause a disaster? That seems like the holy grail.
It really is, and it's a huge challenge. IVUS can give us information about plaque size and remodeling that outward bulging, excessive remodeling might be a warning sign.
Okay.
We can also assess endothelial function non-invasively, like with brachial artery flow-mediated dilation. Impaired function often precedes symptoms and predicts risk.
But what about actually seeing the vulnerable features? The inflammation, the thin cap.
That's where molecular imaging comes in. It's still largely research, but very exciting. Things like MRI with special contrast agents that target macrophages.
The light up inflammation.
Exactly. Or CT with nanoparticle contrast agents doing something similar. Even contrast-enhanced ultrasound using microbubbles designed to stick to VPAM1.
That adhesion molecule.
Yes, to identify active inflammation right on the endothelial surface. These techniques hold promise for identifying high-risk plaques in the future.
Wow. This has been quite the journey through a really complex condition, atherosclerosis. Starts so early, often silent for decades, yet it's this massive global problem.
It really is.
And it's clear it's so much more than just clogged pipes. It's this dynamic inflammatory process involving lipids, immune cells, the artery wall, all interacting.
Absolutely. I think the early origins are often surprising to people, and just how quiet it can be despite the huge impact. And these plaques, they're constantly changing, growing, maybe stabilizing, sometimes regressing.
And sometimes rupturing.
And sometimes, tragically, rupturing. Yes.
So thinking about all this.
Yeah.
The early start, the role of inflammation. It really makes you wonder, doesn't it? What are the most impactful things we can do, maybe starting much earlier in life, to cut our risk before symptoms even appear.
Mm.
Or looking ahead with these new imaging tools trying to spot vulnerable plaques.
Yeah.
How might that change preventative care down the line? Could we be much more targeted?
Those are definitely the big questions. It's a field that's constantly evolving. Hopefully, this dive gave you, our listeners, some valuable insights into just how critical this area of health is.
Absolutely. If this sparked your interest, definitely explore further. There's so much ongoing research into atherosclerosis. It's fascinating and, well, incredibly important for all of us.